Today we will study about the DNA repair process of double-strand DNA repair and single-strand DNA repair. First, we study about double-strand DNA repair, and there are those two processes that happen. One is non-homologous end joining, that is first process, and another is homologous recombination, that is more precise and more accurate process. One is non-homologous end joining. Non-homologous end joining, it brings together two ends of DNA fragments to repair double-stranded break. When double strand of DNA breaks, it brings two ends of DNA fragments to repair. Homology not required, and part of the DNA may be lost or translocated. And second is homologous recombination. Homologous recombination is more accurate and more precise DNA repair mechanism as compared to non-homologous end joining. It requires two homologous DNA duplexes. One strand from damaged dsDNA, double-stranded DNA, is repaired. It uses homologous DNA, double-stranded DNA as a template. It is when this process homologous recombination is defective, it is defective in several breast and ovarian cancer. Defective in breast and ovarian cancers. With BRCA1 and BRCA2 mutation. And in certain types of Fanconi anemia. It restores duplexes accurately without loss of nucleotides. Single-strand DNA repair. One is nucleotide excision repair, second is base excision repair, and third is mismatch repair. Nucleotide excision repairs, it is more specific endonuclease removed oligonucleotide containing damaged bases. Nucleotide excision repairs. This occurs in G1 phase of the cell cycle. This process of nucleotide excision repair is defective in pigmentosum. Nucleotide excision repair. This is defective in Xeroderma Pigmentosum, where inability to repair DNA pyrimidine dimers caused by ultraviolet exposure. It presents with dry skin, photosensitivity, and skin cancer. It presents with dry skin, photosensitivity, and skin cancer. Second is base excision repair. Base excision repair, it basically removes the damaged bases in the base specific, like glycosylase removes altered base and it creates epurenic epidemidinic site. Endonuclease cleaves 5' and removing or more nucleotide. PLiES cleaves 3' and it occurs throughout the cell cycle, and it's very important process in repair of spontaneous toxic deamination. It repairs spontaneous toxic deamination. Mismatch nucleotide in newly synthesized strand are removed and gap is filled and they are released. It occurs predominantly in S phase of cell cycle. It predominantly occurs in S phase of cell cycle. This process is defective in a syndrome in Lynch syndrome, hereditary non-polyposis colorectal cancer HNPCC. DNA repair double strand DNA. There are two processes that happen. One is non-homologous end joining, and another is homologous recombination. Non-homologous end joining, it brings together two ends of DNA fragments to repair double-stranded breaks. Homology is not required in these processes. In non-homologous end joining, part of the DNA may be lost or it translocated. Part of the DNA may be lost or it may be translocated in non-homologous end joining. Homologous recombination. It requires two homologous DNA duplexes. A strand from DNA, damaged DNA, double-stranded DNA is repaired using a complementary strand from intact homologous double-stranded DNA as a template. It is defective in breast ovarian cancer with BRCA1 or BRCA2 mutations and in certain types of Fanconi anemia. Fanconi anemia, this homologous recombination, it is defective. It restores duplexes accurately without loss of nucleotide. It's more accurate and no nucleotides are lost in homologous recombination. Single strand repair. There are three types of single strand DNA repair: nucleotide excision repair, base excision repair, and mismatch repair. In nucleotide excision repair, specific endonucleases removes oligonucleotides containing damaged bases. DNA polymerase and ligase fill and reseal the gap. The gap is filled and resealed by DNA polymerase and ligase. It repairs bulky helix distorting lesion, example pyrimidine dimers. Nucleotide excision repair, it occurs in G1 phase of the cell cycle. It's defective in Xeroderma Pigmentosum. Its inability to repair DNA pyrimidine dimers caused by ultraviolet exposure. It presents with Xeroderma pigmentosum. It presents with dry skin, photosensitivity, skin cancer. And second is its base excision repair. It repair damaged base in the DNA. Damaged base are removed and it repairs small mistake. Base excision repair. Its base specific glycolase enzyme remove altered bases and create AP, apurinic and apyrimidinic site. AP endonuclease cleaves 5' and it removes more nucleotide. PLiES cleaves the primate. DNA polymerase B fills the gap and DNA ligase seals it. It occurs throughout cell cycle and it's very important in repair of spontaneous toxic deamination. Mismatch repair. Mismatched nucleotides in newly synthesized strand are removed and gap is filled and resealed. It occurs predominantly in S phase of cell cycle and it's defective in Lynch syndrome, hereditary non-polyposis, colorectal cancer.